So now, let us go to the electron transport chain. In the ETC, or the electron transport chain, this happens in the inner mitochondrial membrane, which is folded into cristae. This process makes use of the protein complexes, or the cytochrome proteins, and with the addition or with the help of the ATP synthase, which is an enzyme to make now ATP molecules. And in the process, the ETC pumps hydrogen protons across the membrane. Okay. Again, this is where the electron transport chain takes place in the inner mitochondrial membrane. Okay, so again, this is where it happens. It happens in the inner mitochondrial membrane, and the reason why this is folded into cristae is to increase the surface area, therefore increasing now the production of ATP molecules. Now, in the process, again, what are being used in the electron transport chain are the NADH and the FADH2. Now, this figure now presents the oxidation of the NADH. Now, in this figure, the NADH molecules are oxidized, meaning to say they lose electrons. Their electrons are given to the first protein complex in the chain. The electrons now are going to be passed from one protein to another protein until they reach the last protein in the chain. As the electrons pass through the protein complexes, it powers up now the movement of hydrogen protons from the matrix to the intermembrane space, forming now a gradient or an imbalance. Therefore, there is a potential energy. This potential energy is going to be used now to create ATP molecules through the enzyme called as the ATP synthase. As the protons go back to the matrix, the kinetic energy is harnessed by the ATP synthase to phosphorylate now ADP molecules to create now ATP molecules. Also, at the end part of the chain, what happens there is that the electrons are going to be used by the oxygen molecules to be reduced into water to make now the chain flowing or continuous. The produced water now leaves the cell and eventually the body through sweats or urine. Hence, we become sweaty when we work out because of the increased supply of oxygen and its reduction into water. Therefore, this means that oxygen is very important because it acts as the final electron acceptor in the electron transport chain. This is also the reason why a person dies when deprived of oxygen, because the body does not generate ATPs anymore for its metabolic processes because the ETC now does not flow anymore. Okay? This slide now, this one, this slide now presents the oxidation of FADH2. Now, the oxidation of NADH and FADH2 is just the same except to the first protein that accepts their electrons. As what you can see here, the FADH2 immediately donates its electrons to the second protein complex, which is this one. This means now that it can only move lesser number of protons from the matrix to the intermembrane space. So that's what you can see here. As the electrons move across or move through the electron transport chain, it has only moved approximately 3 hydrogen protons. But as what you can see there in the movement of electrons coming from NADH molecules, it has actually moved 6 molecules or 6 hydrogen protons. Therefore, this means now that there is a difference on the number of ATPs that they generate. Now, again, the hydrogen protons move by diffusion from the intermembrane space to the matrix through the enzyme called as the ATP synthase, generating now ATP molecules. This type of phosphorylation again is called as oxidative phosphorylation because it involves now the oxidation of the molecules, which are the FADH2 and NADH, to make now ATP molecules. And in the process, each NADH is equivalent to 3 ATP molecules, while each FADH2 is equivalent to 2 ATP molecules, for the reason that the NADH has moved, or NADH is moving greater number of protons than FADH2. Therefore, For prokaryotes, or those that are having membrane, or for eukaryotes, I mean, or those that are having membrane-bound organelles, the total net yield for coming from one glucose molecule is 36 ATPs. So again, in the FADH2 from the Krebs cycle, we have 2 FADH2 that is going to be used in the electron transport chain. And again, a molecule of FADH2 is equivalent to 2 molecules of ATP. Therefore, this is equivalent to 4 ATP molecules. From 6 NADH, a molecule of NADH is equivalent to 3 molecules of ATP. Therefore, it produces now, or it creates now 18 molecules of ATP, specifically in the electron transport chain. And also in the Krebs cycle, there is also a production of 2 ATP molecules through substrate level phosphorylation. In the grooming phase, there is also a production of 2 NADH molecules, and those 2 NADH molecules are processed in the electron transport chain. Therefore, they create now 6 molecules of ATP. Now, in glycolysis, there is a production also of 2 NADH molecules, and these 2 NADH molecules are going to be processed in the electron transport chain, making now 4 ATP molecules. Now, the reason for this one is going to be explained later on. This should be equivalent to 6. However, in this case, this is only equivalent to 4 ATP molecules. And also, last one, for glycolysis, it has produced a net ATP of 2 through substrate level phosphorylation. Therefore, from the oxidation of one glucose molecule for eukaryotic cells, it can produce at approximately 36 ATP molecules in total. Now, this is the reason why, this is an overview of the process, specifically in eukaryotes. Again, in glycolysis, this is what happens. So in glycolysis, the glucose is broken down into 2 molecules of pyruvates. And also, in glycolysis, there is a production of 2 ATP molecules through substrate level phosphorylation. But aside from that, there is also a production of 2 molecules of NADH. Now, in pyruvate oxidation, these 2 pyruvates are going to enter now the mitochondrion, specifically the mitochondrial matrix. Now, in the process, these 2 pyruvates are converted into 2 molecules of acetyl CoA. And in the conversion, there is a production of 2 molecules of NADH. And these 2 NADH molecules are going to be processed in the electron transport chain. Therefore, this can now generate 6 ATP molecules. Now, these 2 acetyl CoA is going to enter now the Krebs cycle. And in the Krebs cycle, there is a production of two molecules of ATP through substrate level phosphorylation. And in the 2 cycles, it can produce 6 molecules of NADH and 2 FADH2, which are later on again used in the electron transport chain. And again, these 6 NADH molecules is equivalent to 18 molecules of ATP, and 2 FADH2 molecules is equivalent to 4 ATP molecules. Now, these 2 NADH molecules coming from glycolysis are also going to enter the mitochondrion. Upon entering the mitochondrion, there is the expense, or there is expenditure of 2 ATP molecules. 1 ATP is spent in 1 NADH molecule. Therefore, instead of putting here 6, we can only put 4 ATP molecules here. It's because it needs 2 ATP molecules in order for these 2 NADH molecules to enter now the electron transport chain, or the mitochondrion for the electron transport chain. Now, on the other hand, for prokaryotes, or those cells or those organisms in which their cells lack membranes, they actually have a greater amount or a greater yield of ATP. So in total, they get a total of 38 ATPs. So from the 2 molecules of FADH2 and Krebs cycle, this is equivalent to 4 ATPs. 6 times 3, that's 18. In Krebs cycle, there is also a production of 2 ATP molecules through substrate level phosphorylation. In grooming phase, 2 times 3, that's 6. And for glycolysis, that's 2 times 3, that's 6. And then for the still in glycolysis, there is a production of 2 ATP molecules through substrate level phosphorylation. The reason why this is equivalent to 6 molecules of ATP, it is because, again, these cells lack now membranes. So the NADH molecules do not need ATP molecules anymore in order for them to be processed in the electron transport chain. Therefore, this is already equivalent to 6 molecules of ATP. And by the way, for prokaryotes, since they do not have mitochondrion, their electron transport chain happens in the plasma membrane or in the cell membrane. Therefore, there is no need for the two NADH molecules to spend ATP molecules in order for them to be processed in the electron transport chain. Now, in addition to glucose, what other various food molecules are used in cellular respiration? In the food that we eat, we don't only eat glucose molecules, but the energy that we use also come from other molecules around us. For example, the carbohydrates, which are called as the polysaccharides or the complex carbohydrates, for example, for polysaccharides, and also for the fats, for example, the building blocks, which are the glycerols and fatty acids, are also used in the cellular respiration as a source of the production of ATP. And also for the proteins, we have the amino acid.